Alright, these notes are on the cell organelles from Chapter 4, Section 3. The standard that goes along with these is that you need to be able to identify the cell organelles from descriptions, micrographs, and cartoons. So we're going to go through all of those. The cell membrane has three basic functions, and you may remember the simple versions of these from middle school, but we're going to bump it up a level and use some different language. The first one, in middle school you might have heard that it separates the inside of the cell from the outside of the cell. And really what we're talking about here are the chemical reactions that occur inside or outside the cell. It may seem kind of silly, but it's very important that the chemical reactions that go on inside the cell go on inside the cell and not on the outside of the cell. So that's one. The other one that you may have heard is that the cell membrane controls what goes in and out of the cell. And that's true. It's called being selectively permeable. The third one is that it allows the cell to ingest and excrete large particles. So it can take in or release very large particles through the cell membrane. Chapter 5 covers all of these in more depth. So now that you know a little bit about what the cell membrane does, what is its structure? What is it made of? And the first thing that it's made of is a phospholipid bilayer. And that may sound like a big scary word, but it isn't. The first part of the word, phospho, should remind you of phosphates. So there's phosphorus or part, and then lipid, and lipid is another word for a fat. And then bilayer, of course, means two layers. So the cell membrane is actually lots of these phospholipid molecules arranged because of their chemical properties, and they form a nice big sac, kind of like a plastic baggie, and you have the inside and the outside. So here's a picture of what that looks like. These phospho part right here, the phosphorus heads, they're actually attracted to water, which is an important chemical property that they have, and that's called hydrophilic. The tails, each phosphate head has two fatty acid tails, and those are the fats, those are the lipid part. And you guys know that oil is a type of fat, right? Well, lipids are a fat, and you guys know that oil and water don't mix. So this is an important property, that oil and water don't mix. These are called hydrophobic because these fats don't like water. So hydro meaning water and phobic, you guys know about phobias, fears. So since people are mostly, and animals in general, and organisms are mostly made of water, this becomes a very important trait. So if you were to throw a bunch of these individual little molecules into some water, they would actually arrange themselves this way because the hydrophobic lipids will push all of the water that they can away from them, and these phosphorus heads will attract the water. So in order to completely stay away from the water, the phospholipid tails will surround themselves with nothing but other lipid tails and the phosphorus heads, and the phosphorus heads separate them from the water. What you may not have known is there's not actually anything other than this property of not wanting to be around water that holds these things together. So it's very fluid, very dynamic. If you could push your finger in it, you could stir them around, kind of like the Cheerios floating on the top of your milk. You can actually move them around and chase them. If this part were the Cheerio and you had the phospholipids hanging down below. So there's nothing actually attaching them to one another. So this phospholipid could easily kind of migrate and move around between all of the others. Now, I have also called this a plasma membrane because all of the other membranes in the cell that are around other organelles, like the nucleus, the nuclear membrane, are constructed this way. All of the membranes in the cell are constructed this way. So, although we're talking about the cell membrane right now, in the future, whenever we talk about a plasma membrane, know that it's constructed and it looks like this. So the cell membrane has a couple of unique features, one being the transport proteins. Oh, let's talk about this first. Here's the micrograph picture of the cell membrane. Take a moment and look at that and read this. So those transport proteins, this is what makes the cell membrane selectively permeable. These are the things that allow certain molecules in the cell and certain molecules to get out of the cell. It has nothing to do with whether the molecules are good or bad or nice or mean, because molecules aren't necessarily any of those things. It all depends on the chemical properties that those molecules have. And this is how sometimes harmful things like bacteria and viruses can still get into our cell because they happen to have the right chemical makeup in order to get into our cells. The other component is carbohydrates, also called sugars. And these carbohydrates act as little tags to identify the cell. So our immune system functions partially on these tags and recognizes the cell as part of us or not. I mentioned a moment ago that the cell membrane, those phospholipids are not attached to each other. And the way we describe that property is called the fluid mosaic model. Now you're probably familiar with what a mosaic is like in art. This would be an abstract mosaic. It's made up of lots of little pieces. The cell membrane is the same way. If you wanted to say that all the green and yellow ones were phospholipids, you would see the little heads and the tails would be behind. But then the pink ones could be the proteins, the blue ones could be the sugars, and they are all free to move around on the surface of the cell membrane. So unlike a mosaic that's anchored down in tile or something else, this is fluid and dynamic, and each individual little piece can excuse me, can move around inside. Here's what a drawing of the cell membrane would look like. You've got your phospholipid heads and tails here. The blue structures are the proteins, and you can see that the transport proteins are kind of like a straw that's been punched through the cell membrane. It allows molecules to move in and out of the cell. Up here, these hexagon-shaped things are the sugars, the carbohydrates, and these are the tags that identify the cell. The cell membrane also contains cholesterol. Here's a video of what the cell membrane would look like if we computer animated it.
Between the living machinery of the inner cell and the harsh conditions of the outside world stands the cell's plasma membrane. As crucial as this barrier is, it's surprisingly flexible. Push it and watch it move. Poke hard enough and it might break and begin to regroup. The lipid molecules of the membrane naturally assemble in a double layer because their tails repel water as their heads attract it. Throw in some cholesterol and a few carbohydrates, and you have the basic structure of a plasma membrane. Within these lipid molecules, we also find different proteins which do various things for the cell. For instance, they receive signals from the world outside, or they transport nutrients and waste. So nature composes the membrane with a combination or mosaic of different lipids, carbohydrates, and proteins. And these molecules are not stationary. They constantly move within the structure, fluidly changing their positions. The survival of all life rests on this veil of material. A supple membrane, just 2 molecules thick.
All right, let's go back to our slideshow. So that's the basics of the cell membrane. We'll actually cover it in much more detail in Chapter 5. Let's talk briefly about the mitochondria and the chloroplasts. Mitochondria makes energy, or ATP, from glucose in the cell. Mitochondria has an inner and an outer membrane that again is made up of those plasma membranes of the phospholipid bilayers. Here's a picture of a mitochondria. So you can see the outer membrane. You can also see the inner membranes here. These inner membranes are where the chemical reactions take place to make the ATP. So the more membranes there are, the more ATP that mitochondria can produce at one time. You'll also notice an identifying feature of the mitochondria being these little black dots. And that's because mitochondria do something that no other cells do. And that is they reproduce themselves, or no other organelles do, and that is that they reproduce themselves. They have their own DNA, and it's kind of interesting. So there you go. Here's another picture that looks quite different. This one is long and kind of skinny, and it doesn't have a whole lot of the membranes inside. This one, however, is kind of big and fat and has lots of inner membranes, but it also has these dark spots, which is an identifying feature of the mitochondria. Chloroplasts are where photosynthesis takes place in order to make glucose. A quick note here. Plant cells have chloroplasts to make glucose, or sugar. After they make this sugar, plant cells do have and need mitochondria to turn that sugar into ATP. It's a common misconception, especially coming out of elementary school and middle school, that plant cells have chloroplasts and animal cells have mitochondria. But plant cells also have and need mitochondria. So let's take a look at the chloroplast. It's a round organelle. Here's the drawing. You have stacks of grana, which in the micrograph picture are these squares. You have this string-like or spaghetti-like substance that wraps around those stacks, and you can see that here in the micrograph picture. So this is what a chloroplast looks like under a microscope. Ribosomes. They're found free-floating in the cytoplasm or attached to the ER, the endoplasmic reticulum. I'll make a note here that ribosomes themselves do not have a membrane, so they are found in prokaryotic organisms. However, prokaryotic organisms do not have an endoplasmic reticulum, which is almost entirely membrane. So what do ribosomes do? Ribosomes synthesize or assemble proteins, kind of like you can assemble a Lego ship or some other sort of Lego castle, whatever it is that you like. You don't actually make the Lego blocks. You just take them, look at the instructions, and put them together. And that's what the ribosomes do. They have a set of instructions, and they take the building blocks of the proteins and simply put them together. So where do those instructions come from? They come from the DNA in the nucleus. Here's a picture. Most of the time ribosomes are drawn as just tiny little dots, because in reality that's what they look like, tiny little dots. In this hand-drawn picture of the cell, these little tiny blue things would represent the ribosomes. They're the smallest organelles in the cell. And here it's showing them on the endoplasmic reticulum, which is often attached to the nucleus. Here's the micrograph picture. The endoplasmic reticulum is this long, sort of maze-like structure of membranes, and the tiny little dots that are attached to it are the individual ribosomes. Vesicles are another interesting organelle, and a vesicle is a general term for any organelle that is just a membrane bubble. Now, when you see a micrograph picture of a cell or a drawing of a cell, most of the time you're just going to see a round bubble. And you're going to call it a vesicle because you can't tell what's inside. And what's inside is what makes it either a vacuole or a lysosome or some other organelle. So let's talk about the vacuoles. They're storage for water and chemicals, and in plant cells they can take up 50 to 80% of the cell space. Other single-celled organisms often have vacuoles as well. They're not unique to plant cells. Lysosomes contain digestive enzymes. Lysosomes are pretty cool. Our immune system, white blood cells, go around and they ingest bacteria and other harmful things. So how do they get rid of it? And the answer is these digestive enzymes. In fact, these digestive enzymes are so powerful that if the lysosome were to break open, it would digest the cell. So here's a micrograph picture of the Golgi apparatus. So it looks like a stack of membranes here, and all of these little circles coming off are vesicles. Now we don't know what's going to be in them or what's in them now, so we just call them vesicles. Perhaps there'll be a lysosome, maybe not. I want to show you something important about the vacuole here. When you see a hand-drawn picture of a plant cell, the vacuole often appears kind of small. The reason for that is they want to show you all of the other organelles too. When you look at a plant cell under a microscope, however, the central vacuole may take up so much space that it squishes all of the other organelles to the side to the point where you may not actually be able to tell what they are. In this one, you can see a couple of other vesicles. We don't know what's inside them, so perhaps they're lysosomes, maybe not. But here are some other vesicles, and here's the central vacuole. The last organelle we're going to talk about is also free of membranes, which means it occurs in prokaryotic cells as well, and that's the cytoskeleton. The cytoskeleton is a network of tubes and filaments, microtubules, and they're kind of like cables that fill up the inside of the cell. They have a couple of key functions. One, intercellular mobility. It moves vesicles and other organelles from one side of the cell to the other. It also helps split the cell during mitosis. The last thing that it does is this is the framework that gives cells their basic shape. It's tempting to say that the cell membrane gives the cell its shape, but really, again, the cell membrane is kind of like a plastic Ziploc bag. The cytoskeleton is what pushes out from the inside to give it its shape. Your book does a really good job of going into all the different types of fibers and filaments and things like that, but really they're all part of the cytoskeleton. And I'm not too concerned that you know the individual types of fibers and filaments. Here's a micrograph picture of the cytoskeleton. It's kind of cool. All of these individual little cables are microtubules that can move things throughout the cell. The flagellum and the cilia are made of similar things to the cytoskeleton, except they occur on the outside of the cell. The flagellum, and hang on. The flagellum is a tail-like appendage used for locomotion, and the cilia is a hair-like appendage also used for locomotion. Here's hand-drawn pictures. This is a euglena, and it has a tail to move itself around. Here is a paramecium, and it is covered with hair. When we draw the cilia, we usually just draw it in a circle around the cell, but in fact it covers the entire surface of the cell. So let me show you a micrograph picture of that. Here the cilia covers all of the cell, not just the little rim like we would draw. And here's the flagella. There happen to be 3 on this bacteria. Now, with your table, look at the last 3 organelles that I haven't covered. The nucleus you should be pretty familiar with. The Golgi apparatus, also called the Golgi complex, is pretty straightforward and easy to learn on your own. The endoplasmic reticulum has 2 types, the rough and the smooth ER. So look those up now with the time remaining in class.